right, so we are moving on to chapter 16, which is on glycogen metabolism. So again, we're going to learn a lot of metabolism from a very carbohydrate-centered viewpoint. So again, we've learned about glycolysis, and now we're going to think about potential sources that we have for the glucose for glycolysis. So again, glycogen is also called animal starch, again, your way of storing glucose. So in this lecture and this lecture's activities, we're going to have some times where we kind of review glycogen and glucose structure and kind of think about how that structure is going to relate to our ability to use that as a fuel. We'll kind of review some overviews kind of metabolism and think about branch point metabolites, and then we'll actually delve into the details of kind of a three-step process to think about for glycogen breakdown. But as we kind of see here, we're going to focus on glycogen breakdown today. We'll kind of introduce the concept of glycogen synthesis, but as we do with all of our metabolic pathways, we'll think about control of glycogen metabolism, and then we'll think about kind of two other processes, which are gluconeogenesis, which is the reverse of glycolysis. You can think of it as running glycolysis in reverse, and then kind of some other carbohydrate pathways. So again, if we're going to be thinking about synthesizing carbohydrate polymers, we'll think about some other important glycosidic linkages that we might think about creating. And I know this is a really busy slide. Don't try to read necessarily everything that's on it. This is a lot of information that we will go through in our activity during this class's activity. We're going to review the structure of glucose, review the structure of glycogen. Again, we learned those in Chapter 8 in Biochem 1, but we're going to review them and make sure we understand all the important components. So again, starting with glucose, remember that glucose is an aldose sugar, and when we cyclize it, that's a way that we kind of ensure that we are protecting that aldehyde functional group. So again, converting it into a hemiacetal. So this one carbon here is very important. We refer to that as the anomeric carbon. And again, when we tie this together in a ring form, we do protect the carbonyl, but if there is a free OH on the anomeric carbon, we refer to this as a reducing sugar. And remember, we referred to that in that way last semester when we learned about what reducing sugars were, is because we were one step away from having a reactive carbonyl. All we need to do is ring open this sugar. We've got a reactive carbonyl that then could do some redox type chemistry. Again, this is beta D-glucose, beta because this anomer has the OH on the same side as the exocyclic CH2OH group. Again, beta D-glucose being nature's favorite sugar. So again, just some review. We'll go over this during our activity and our review of structure tomorrow. Some other things then to think about is when we polymerize glucose. So kind of starting down here and kind of looking at this little image here, and we'll have stuff that we do with this in our activity tomorrow. When we just link glucose together with alpha 1,4 glycosidic bonds, and hopefully this is starting to ring a bell with stuff from last semester, we would make a linear polymer. Each one of these little spheres here represents a glucose molecule. Again, the line here represents the glycosidic bond that would be connecting them, and again, that would connect the anomeric carbon on C1 of one sugar to the C4OH group on another sugar. That's how we make that alpha 1,4 glycosidic bond, is we essentially have the these two carbon atoms hooked together. And we went through that chemistry. We'll actually look at some mechanisms to think about that. But if all we did was create alpha 1,4 glycosidic bonds, we would have a linear homopolymer of glucose. It would have one kind of back end of the molecule, which would be called the non-reducing end. It's an end, so we call it an end, and it's non-reducing because on that glucose monomer, the anomeric carbon would be tied up in this glycosidic bond right here. However, if we look at the other end of the molecule, we would call this a reducing end because this sugar would have its C4 group tied up in this glycosidic bond, but the C1 carbon would not be tied up in a glycosidic bond, and so we'd refer to that as the reducing end. So when all you have is a linear homopolymer of glucose that's alpha 1,4 linked, you have one reducing end and one non-reducing end. Okay, What we'll learn about with glycogen metabolism is that enzymes chew in from non-reducing ends. And if all you have is one end of the molecule that you can start mobilizing glucose from, that's a very slow process. So the purpose of branching is that we introduce more non-reducing ends. And if this is confusing right now, we'll go through it more in our activity in class. Okay, but what we have is, remember, anytime we want to make a glycosidic bond, we just have to take an anomeric carbon and we're going to hook it up to another OH group. If that OH group happened to be from a C4 on a neighboring glucose molecule, we'd make an alpha-1,4 glycosidic bond. But there's lots of other OH groups, and in particular, the C6 exocyclic CH2OH group is one that we can utilize. And when we do that, we introduce what we call an alpha-1,6 branch point. So again, we can kind of see that with our little cartoon here. What we'd have is we'd have an anomeric carbon here. Let's imagine that this kind of isn't there. When we have this kind of carbon, and well, I guess let's imagine that that's there. We're going to use the C6OH group from this guy. So here's another little homopolymer down here, a much shorter one. We've got a non-reducing end and then a reducing end. If we take the anomeric carbon's OH group from this guy and we hook it up to the C6 exocyclic OH group of this guy, we create a branch point. What that does is eliminates a reducing end. That's not an end anymore because it's connected here, but it introduces another non-reducing end. So every time we branch, we introduce the number of non-reducing ends. And again, our activity is going to go through that a lot more. So what that ends up creating in terms of a structure of glycogen is we've got a linear polymer of alpha-1,4 linked glucose residues that have alpha-1,6 branch points. And again, we'll talk about the importance of the spacing of those branch points, how long the branches are. All of that plays a lot into how we metabolize glycogen and mobilize glucose. All right, so two terms that we refer to a lot are this concepts of time of plenty and time of need. Again, time of plenty, when your blood sugar levels are high, we call that times of plenty. What that's going to mean for glycogen is we're going to decrease glycogen breakdown and we're going to increase the synthesis of glycogen. Remember, to a point, you can keep about a 12-hour supply of glycogen in your liver and your skeletal muscles, but it's not an unlimited supply like our adipose tissue is. So again, we'll store excess glucose as glycogen to a certain point, but then once we've filled up those reserves, the excess gets converted into lipids, and we'll deal with how that process happens in a few chapters. But again, times of need, if your blood glucose levels drop, then we need to increase the breakdown of glycogen to mobilize glucose to increase and remedy that low blood sugar level. And not surprisingly, then we're down-regulating these synthesis processes. So a lot of information there, lots that's written there, but again, this is going to become part of what we work through in our activity tomorrow during class. All right. So a nice slide here that we can use to summarize a lot of what we've talked about already, right? We've talked about the glycolytic pathway, right? That was chapter 16. We're going to learn about gluconeogenesis here in chapter 16. So again, that's basically taking glucose, or the first phosphorylated form, glucose-6-phosphate, and taking it all the way through glycolysis will generate pyruvate. Taking pyruvate and making it back into a glucose molecule is gluconeogenesis. We'll learn that at the end of this chapter, right? We talked about the fates of pyruvate, right? We're going to consider from this point on really only aerobic fates. We talked about anaerobic fates in chapter 15. And again, noting that this is a one-way arrow, by converting pyruvate into acetyl-CoA by pyruvate dehydrogenase, that kind of commits things to the citric acid cycle down here. And again, if we're going to mobilize fats and kind of convert them into energy, they can only be converted into acetyl-CoA. And since this arrow is one way, what that really means is things that are derived from fats and made into acetyl-CoA cannot be made back into glucose. So, big picture, and I think you guys have gotten this through your head, fats cannot be made into sugar. We'll talk about protein metabolism in a few chapters again, but certain glucogenic amino acids, so certain amino acids that can be derived or made into pyruvate, can be made back into sugars. So again, proteins can be converted into glucose. Some amino acids from proteins can. But again, this is a very taxing process on your body. There's a lot of metabolism that happens to convert those amino acids that happens in the liver and kidneys. We'll talk about that in our protein metabolism chapter. But again, not only is it taxing on some organs, but it's very costly. Again, we talked about fats being able to pay that cost. That's the purpose and the intent of the Atkins diet. Okay, we learned about glucose-6-phosphate, right, in glycolysis as kind of hexokinase converts glucose into glucose-6-phosphate. One of the things that we'll see with several molecules that have arrows essentially coming in and out here, they're what we call branch point metabolites, and glucose-6-phosphate is a really important branch point metabolite. And as we'll see in this chapter, we'll see a lot more that feeds in or out of the glycolytic pathway through this molecule. So again, glycogen being broken down ultimately gets converted into glucose-6-phosphate. And again, if we're going to synthesize glycogen, we begin with glucose-6-phosphate. Just highlighting some other things that are written here. Glycogen, again, is stored in your liver and your skeletal muscle. The importance, though, of the skeletal muscle glycogen is that it's what we call a localized source. That's only going to be used by the muscles. It's not something that can be used to remedy a drop in blood sugar. The liver, however, because when it mobilizes its glycogen, it can deliver it out to the bloodstream, so it's what we call a global source of glycogen. If you hear the term glycogenolysis, so glycogen and then olysis referring to the breakdown, this is the scientific term referring to the breakdown of glycogen, and again, we'll deal with glycogen synthesis after we first deal with breakdown. All right, so having glucose-6-phosphate, right? Glucose-6-phosphate, if we get rid of that phosphate, can be exported and circulated in the bloodstream. And a really important thing to remember, only free glucose can be exported and circulated. So if we want to have that glucose kind of circulate in the bloodstream, we first have to dephosphorylate it, which means we kind of lose that first benefit of that first ATP addition. So it's not going to be worth as much in glycolysis because we'd have to kind of lose that initial phosphate that we put on. But again, only free glucose can be exported and circulated. The last little piece kind of on this very busy slide here is to think about something that we've already talked about. Glucose-6-phosphate can be siphoned off of glycolysis and be made into ribose 5-phosphate. That happens again in the pentose phosphate pathway, which we learned at the end of chapter 15 on glycolysis. Right? So ribose 5-phosphate or the pentose phosphate pathway is important for two purposes, right? Synthesizing NADPH for reductive biocurrency that we use for biosynthetic processes, and also for the synthesis of nucleotides, right? That's what ribose 5-phosphate is used for. All right, so a lot of information I kind of threw out there. We did it very quickly, but again, this is going to be a figure that we'll dissect again in our activity tomorrow, and we'll think about how we use glucose and glucose derivatives in our body. All right, lots of circles that are on here, and unfortunately, because this was annotated on my PC, we can't see that these are actually different colors. So what we have here is I tried to kind of highlight and circle anything that is a non-reducing end here, which we've got kind of highlighted with these arrows, are circled in one color. Anything that's kind of between different things here, we're going to have as yellow. Okay, I think these ones are going to be green, these ones are going to be yellow, and any ones like we see right here, which are branch points that have 3 glycosidic bonds kind of as part of them, are purple. And those colors will make sense in just a minute. But I wanted to kind of highlight when we look at the global structure of glycogen here. So again, we've got alpha 1,4 linked glucose residues with alpha 1,6 branch points. And here again is our one reducing end that we'd have on our glycogen molecule. So again, one thing to think about is every single glycogen polymer is only going to have one glucose residue that is technically a reducing end, because every time we take a branch and we hook it up and make a alpha 1,6 linkage, we use up one of those reducing ends by tying up that anomeric carbon in a glycosidic bond. Okay, so these colors again that I just kind of mentioned, these reducing end or non-reducing ends, excuse me, being green, these kind of intermediate areas, whoops, they're being yellow, and then these branch points being purple. Okay, so we'll see that kind of on our next slide. So a couple of things just to kind of think about here. When we talk about glycogen, it's really what we call glycogen granule. So basically there's about 120,000 residues of glucose that are all kind of hooked together in this branched polymer. And these glycogen granules not only contain these glycogen molecules, but they contain all the synthetic, degradative, and regulatory enzymes to kind of facilitate glycogen metabolism, kind of an all-in-one package. And liver, the dry weight of liver can actually be about 10% of these glycogen granules. Not as much in muscle, but again, muscle's use is tended to be local, whereas liver will provide glycogen and then glucose for global use. All right, so let's flip to the next slide here. Again, these are figures that we will see when we examine structure and think about glycogen breakdown in our class discussions. 3 steps that I want you to think about when we think about glycogen breakdown: mobilization, debranching, and then mutation. So not to say that these are happening completely sequentially, but what mobilization is we have an enzyme that chews in from the end of our molecules, our non-reducing ends. Well, that chewing in can happen until you reach a branch point. So not surprising, the next thing that happens is debranching. Okay, well, once you've debranched, now you have longer ends again, you can go back and you can continue to mobilize some more. Well, what we'll see here as we look at this, what we're going to mobilize is glucose 1-phosphate. And if you remember from what we saw a few slides back, what we have here is glucose 6-phosphate that we get as the end point of glycogen breakdown. So what that means is the glucose 1-phosphate we get for mobilization needs to be mutated to glucose 6-phosphate. All right, so now we're going to go through these steps individually here. The first enzyme that's responsible for doing this mobilization is called glycogen phosphorylase. This is kind of the be-all, end-all important molecule in glycogen metabolism. So this is the enzyme we're going to study regulation of. This is the rate-determining enzyme of this pathway. So we'll look at the chemistry that this enzyme undergoes, but again, you're going to have glycogen, that's a polymer that's a certain number of residues long, and what's going to happen is we're going to break that glycosidic linkage. Previously, we've only considered hydrolysis of this glycosidic linkage. What we're going to consider for glycogen now is phosphorylasis. So we'll look at that from a mechanistic standpoint, and we'll see how water would participate in this process, and then we'll see how phosphate could take its place and participate. But what that really means is we don't generate free glucose, we generate glucose with a phosphate where an OH normally would have been, so glucose 1-phosphate. So again, we're going to look at the details of this enzyme, but I want you to understand that it's a rate-determining enzyme of glycogen breakdown. So again, it rapidly mobilizes glucose. About 50% of the glycogen is rapidly mobilized as glucose 1-phosphate by this enzyme doing its job. What must happen in order for this enzyme to work is residues must be at least 5 units away from a branch point. So what we can see here is we could have glycogen phosphorylase kind of chomping onto this non-reducing end, and it would chew in, but it would have to stop right about here because we are within 5 units of a branch point. And then we'll learn how this glycogen debranching enzyme works. But that's glycogen phosphorylase. All right, I promised you a mechanism. All right. So again, we'll work through in class looking at the hydrolysis of an acetal to a hemiacetal. Remember, acetals represent the glycosidically linked glucose or sugar molecules, and then breaking that glycosidic linkage will give us to the cyclized sugars, which again are hemiacetals. So if we think about this from a mechanistic standpoint, right, this acetal here would represent a cyclized sugar. This OR linkage would be our glycosidic linkage. We protonate that linkage to make a good leaving group. Great internal nucleophile that's going to allow us to eliminate that good leaving group. Generate a very electrophilic intermediate that can be nucleophilically attacked by water. Again, deprotonation of water, either as it's incoming or afterwards, is going to leave us with our hemiacetal, again, being our cyclized sugar. Now, I apologize, we don't have different colors here. If we consider the phosphorylasis of this, so that is, rather than this nucleophile being water, if this nucleophile is phosphate, like inorganic phosphate, okay, when we have this nucleophilic addition here, what's going to happen is that we're going to have an O-phosphate group rather than an OH group here. So that means we're going to have a phosphorylated sugar at the 1 position. And again, that's how we generate glucose 1-phosphate. All right, we're going to look at the enzyme active site here. We're going to learn about a new B vitamin that we need, vitamin B6. Vitamin B6 is a prosthetic group that we use for pyridoxal 5-phosphate. So PLP is going to be this group that we see structure shown right here. We've got an important aldehyde functional group here that's going to covalently attach to a lysine within the enzyme active site. You guys should know the chemistry that's involved between an aldehyde and a primary amine like we see with lysine. So again, we're going to have this PLP that's within the enzyme active site. And you might be thinking, hey, this is really cool. We've got glycogen. We're going to have phosphorylases chemistry. So this must provide the phosphate as a nucleophile. No, what really happens here is PLP is not a source of phosphate. Rather, it actually serves as an acid-base catalyst. So I know it would be real easy and simple to think about it serving in that role, but that's not actually what happens. It truly is inorganic phosphate that comes in and serves as the nucleophile in this reaction. Okay, so let's actually look at this enzyme active site. Again, we'll spend some more time in class focusing on this a little bit. Enzyme active site, right, we're going to have glycogen is going to bind with the co-substrate inorganic phosphate. Here's the enzyme active site again, where we're going to have our PLP that's covalently bonded. Again, the phosphate is the only part that's really important. But again, that phosphate is serving as a general acid-based catalyst. So what we can see here is we've got this guy that's going to act in a general acid catalyzed fashion. But if we start again where our arrows show, we can imagine that we can have cleavage of this alpha-1,4 glycosidic linkage, which is facilitated by inorganic phosphate here acting as a general acid, and its acidity is facilitated by PLP acting as a general acid. So again, we've got that cascade there that sets up to facilitate the protonation again of our glycosidic oxygen, our leaving group, and again its departure is facilitated by those electrons on that ring oxygen. We end up with an intermediate here that looks very similar, hopefully, to something you remember seeing last semester when we talked about oxonium ion intermediates. We saw that in, I'm not going to give you the answer, we'll talk about it in class tomorrow. But again, we have a very electrophilic intermediate now that's very susceptible to nucleophilic attack by our inorganic phosphate. And again, the nucleophilicity of that inorganic phosphate is facilitated by PLP acting as a general base. And that's useful because that regenerates PLP in its active state. And then we generate a glucose or a glycogen residue that's shortened by 1 unit, and then glucose 1-phosphate. So again, practice going through your describing enzyme active sites, saying the residue, acting as a mode of catalysis, serves to, what does it do, tell me what it made. All right, so we've mobilized as much as we can until we get within 5 residues of a branch point. So again, here's our branch point here. We've cleaved that 5th residue from a branch point, and we have left what's called a limit branch. So glycogen debranching enzyme is a bifunctional enzyme. We're going to see lots of bifunctional enzymes, which means it's an enzyme that actually can perform 2 very different catalytic functions. One of the functions it has is called an alpha-1,4 transglycosylase activity. Like the name kind of implies, we've got a cross and then movement of this glycosyl unit. So we're going to be moving this limit branch. And what's kind of interesting is it moves not right up to where the branch point is, but 1 away. So this entire thing is called a limit branch, but we move 1 residue away, and we move this to the end of another limit branch so that we create a longer piece that's now available for glycogen phosphorylase to chew in on. Okay, but then we have this little green nub that we have left here. What we have now is the second activity of the glycogen debranching enzyme that's going to facilitate this process, and this is a hydrolysis process. This is truly a hydrolysis process. In this case, we're going to have water that nucleophilically attacks and helps cleave this glycosidic bond, so this green residue really is liberated as free glucose. I'm going to flip back just for a second, and we'll deal with this more when we do an activity in class tomorrow, but when we consider looking at these pictures, right, and we consider that this purple residue represents a branch point, we're going to have a yellow residue that's adjacent to that branch point that is going to be able to be liberated as free glucose. Everything else is glucose 1-phosphate, so we'll have to go through the next step, but about 10% of the residues are going to be these little nubs that are going to be able to be liberated as free glucose. Everything else, glucose 1-phosphate. Okay, so our debranching enzyme is going to kind of work in concert with glycogen phosphorylase to ultimately give us a pile of about 10% free glucose, which is ready to do whatever, and then a pile of 90% of glucose 1-phosphate. So the last thing we need to do is make that into glucose 6-phosphate because that's the branch point metabolite that's useful. So again, only G6P is able to be used in glycolysis. And again, that's kind of useful in muscles because when you mobilize glucose from glycogen stored in your muscles, you generate it as glucose 1-phosphate, which in those situations means it bypasses that first energy input of ATP. So you actually get more ATP out of glucose or glucose 6-phosphate that's derived from glycogen than if it were to be free glucose because you bypass that first step. However, if you're the liver and you're going to be transporting this out to the bloodstream for other use, you actually lose that benefit because you have to dephosphorylate it. Only free glucose can be exported and then circulated in the bloodstream, so there's a phosphatase that can actually snip that off and facilitate that process. However, we need to get to G6P because the endpoint of phosphorylasis by glycogen phosphorylase is G1P. Okay, so we've got phosphoglucomutase, which basically is going to take G1P in the enzyme active site. There is a serine that has a phosphorylated OH group on it. That's going to transfer that to the C6 group. We get a glucose 1,6-bisphosphate intermediate, and then we transfer that to that phosphate from the 1 position back to the serine, regenerating it in the enzyme active site, and then leaving G6P as our product. Again, I'm not going to tell you the answer to this. Think about it. We've seen an enzyme before that operated by doing mutase. Remember, mutase enzymes transfer whole functional groups? We've seen that before. We've seen an enzyme that transferred a phosphate group and made a bisphosphate compound and then took the other one off. So think about what that is. We'll review that tomorrow. All right, so to summarize, glycogen got mobilized into G1P by glycogen phosphorylase. We had to move branches in order to create longer pieces that glycogen phosphorylase could continue to mobilize. And again, that debranching enzyme not only moves those limit branches, but it's going to remove those nubs and give us that free glucose. And at the end of the day, that 90% pile of glucose 1-phosphate gets made into glucose 6-phosphate. All right, so we're going to spend some time talking about how we regulate glycogen phosphorylase. So importantly, glycogen phosphorylase is regulated by covalent modification. We've already learned this. Remember chapters 12 and 13 when we learned about kinetics and then we talked about cell signaling? We learned about the importance of phosphorylating phosphorylase. Remember that? Okay, so we're going to revisit that and we're going to begin kind of putting all of these pieces together. So again, phosphorylase is subject to covalent modification, right? We've got the phosphorylated form, that's the A form, is the more active form, but that's not always the case. But for glycogen phosphorylase, if we phosphorylated it, it's active. The dephosphorylated form is less active. But again, we learned about that stuff in chapters 12 and 13. Remember, glycogen phosphorylase was that enzyme that not only was subject to covalent modification, addition or removal of a phosphate, it was also subject to allosteric regulation, so changing between T state and R state. Do you guys remember what determined whether phosphorylase was T state or R state? I'll give you a hint. It has to do with N-terminus. See if you can remember that. We'll review it in class tomorrow. And continue to keep thinking about activators and inhibitors. All of our activators and inhibitors should make sense. Things that are global inhibitors for enzymes like phosphorylase would be ATP, G6P, glucose. These are fed state signals. AMP is a need state signal. Start making a list of these so that we remember what they are, because these things don't just act on the single enzymes that we're learning about. They act on a host of other enzymes that help kind of all throughout our metabolic processes. So again, that kind of summarizes or concludes what we want to think about for the 3 steps for glycogen breakdown. So we've got glycogen phosphorylase that mobilizes, we've got debranching that happens with our glycogen debranching enzyme, and then mutation that happens with our glycogen mutase enzyme. So the last thing we need to think about then, I'm just going to introduce, is this concept of glycogen synthesis. So the processes of glycogen breakdown and glycogen synthesis comprise what we call a giant thermodynamic cycle or substrate cycle, right? And we can't have things be downhill in both directions. So again, if it was energetically downhill to take glycogen and break it down into glucose-6-phosphate, we need to have it be the only way it can be downhill or favorable to kind of go in the opposite direction is if we see the input of energy. And this is kind of a theme that we will continue to see, right? We already learned about glycolysis. Glycolysis going in the forward direction went downhill on energy. We gave off energy in the form of ATP. So if we want to do gluconeogenesis and run that pathway in reverse, we're going to need to put energy in. So not surprisingly, we're going to see some kind of parallels to reversing these steps. So if glycogen phosphorylase was responsible for kind of breaking apart glycosidic linkages, we're going to have glycogen synthase, which makes them. If we had an enzyme that got rid of branches, we're going to have an enzyme that puts back branches. And one enzyme that's kind of similar between both pathways is this phosphoglucomutase enzyme. So again, that enzyme is responsible for interconverting G1P and G6P. But again, we've got a low energy compound that we need to create a glycosidic linkage. And the only way to do that is to activate this so that it has a decent leaving group. So again, we'll revisit this in our next lecture, but just wanted to introduce the concept of glycogen synthesis.